Ladies and gentlemen, I think we are about ready to get started once again. Thank you so much for staying with us. I hope you enjoyed lunch, and we're looking forward to a very interesting afternoon session. Before I turn it over to our very esteemed chair for this session, Professor Paul Tambaya. Thanks for coming in, Paul. I do need to note that, unfortunately, Dr. Raja Dar, who is one of the directors of APACI, won't be able to present this afternoon. As such, we will have a little bit more time for the presentations and for questions for the three presentations we have in this session. So please do prepare questions during the presentations. We look forward to them. I will turn it over now to Professor Tambaya. Thank you.
Thank you very much, John. So we're now at session 3, vaccines for the lifespan, combinations, enhancements, and what the future holds. And our first speaker to kick off this session is none other than my good friend and colleague, Dr. Leong Hoe Nam, who is a senior consultant infectious disease physician at Mount Elizabeth Medical Center in Singapore. He is also my go-to person. If anyone is too tired of waiting in the public healthcare system, I usually tell him, please call Dr. Leong's clinic. So he's got a tremendous amount of clinical experience, and he's going to talk to us about the future of respiratory virus prevention, burden, coexistence, and vaccine innovation. Ho Nam.
I just a little bit, so we're not really in a rush for time. So Paul Tambaya, Professor Paul Tambaya is an incredible man. Not only was he inspiring, he was one of the few people who just endorsed me to do infectious disease physician. So he was very much my teacher. Yes, despite me looking so old, yes, he was my teacher. And I still remember the conversation. We didn't have the chance to do an interview. And he said, we'll do it over the phone. Ho Nam, do you know infectious disease is really tough? You have no time for your family. I said, yes. Are you sure you're in for it? Remember the conversation? And he says, yes. So I became an ID physician. And all thanks to Paul. And Paul has been incredible. He's been teaching me on many, many things. On top of that, when I need some services in the public health sector, Paul is my go-to guy. And lastly, a lot of the things about talking and presentation, actually I learned from the great speaker himself, Paul. As you have heard, Paul is just an incredible speaker. So I'm going to talk a little bit about the future of respiratory virus prevention, burden coexistence, and vaccines. And this one is about the future of virus prevention. So it's going to be a little bit of a light touch and go. I do speak for Moderna, and I've received an honorarium. They're very, very nice to put me in a nice hotel and then send me over here. And I get to enjoy the wonderful Hong Kong hospitality and the wonderful Hong Kong people and the wonderful Hong Kong food. Okay, so this is Hong Kong. Great travel hub, connected city, and you're talking about number of visitors, 49.9 million, many coming from mainland visitors. Tons of flight daily. I have it as 1,100. That's incredible. More than 220 global destinations. And in fact, Singapore is often compared with Hong Kong, and Singapore emulates to be like Hong Kong. But Hong Kong has the big brother China at the back and helps a lot. But fortunately, airport hubs are an issue. Airport hubs are an issue because they actually transmit viruses. We learned it from SARS, we learned it from H1N1, we learned it from COVID as well. Very much when it comes to air travel, it transmits viruses. You see, the viruses have become very, very smart, and they travel along with the planes. Passenger travel doesn't help because you carry the virus, transmits over. We can put up all the fever screening there is, but it doesn't really help. You can be in an incubation period and spread thereafter. Vibrant Hong Kong, incredible MTR systems, your buses, and the link between the public transport and infectious diseases, it spreads all quickly throughout. Great transport system for the humans, great transport system for the viruses. So we also know that in terms of viruses, with availability of PCR, we're beginning to understand some of the seasonal variability. So we have the flu viruses in the winter viruses. We have the influenza, we have the RSV, we have the COVID viruses. But COVID is still pretty much designed by the new sequences that comes along. I'm sorry it's getting out of shape because I think it is an Apple thing. I used this on a PC and the PC was fine, but the Apple system just messed it up. We have the adenoviruses and human viruses, which goes out throughout the years. We have the parainfluenza viruses that comes rather at different times, some in October, November, some in March and April. And with the availability of PCR, I begin to understand a lot more about this. This is great. We can learn to predict. We can learn to anticipate. And then we can even learn to try to see if we can do other vaccines available. We have the human seasonal coronaviruses that goes around all throughout the whole year. This data from Singapore. Singapore was in COVID. We were locked down. And thereafter, we had many different viruses coming out. One of the earliest to come out was actually influenza. But we also had the RSV. RSV came along, and then it started coming up with huge numbers. This was in 2023, where the influenza that peaked. Singapore is actually a bit unusual. We're just at the slightly north of the equator, but we pretty much follow the southern hemisphere peaks. But we still have two peaks, one is the smaller one in December. We have COVID-19 as well, and COVID-19, as I kept saying, is here, it stays, it's here, it stays. It's like a nightmare that refuses to go away. Year-round, 2023. This is Korea, Hong Kong, as well as Japan. We have this. The blue actually indicates the winter months. I lost my snowflake. There are snowflakes indicated for the blue columns. The blue columns indicates the winter seasons. And you see that the winter seasons come out together with the RSV as well as the influenza. The red lines is that of COVID. This is Japan. Japan is here. Japan at the top, you have the COVID-19. You have the peak going up, but that's because due to a new COVID strain. You have the influenza here, which parallels very nicely with the colder months. But surprisingly, for RSV is actually in the middle of the year. And apparently, RSV actually transmits quite well when it's warm, humid, with a big air droplet. And unfortunately, for many of these viruses, the symptoms are very similar. For a practicing clinician, I get to see, in private practice, I get to see people with cough, runny nose, sore throat on day one of illness. Great, I can do a swab test, a rapid test. And if it's negative, I know it's not RSV, adenovirus, influenza, COVID, etc. But unless I do the expensive PCR, I can't get the answer. And it's very, very difficult for me as a clinician to try to guess what it is. We do have epidemiology data coming on a weekly basis, telling me which are the circulating strains. And with that, I can try to guess a little bit. But if you actually look at all these symptoms, it's ridiculous to try to estimate or try to guess what the person has. And I thank God for the ability of the rapid antigen detection testing. That helps tremendously. And if the PCR prices fall, it'd be great. And if you look at all the common viruses from flu, RSV, human coronavirus, human rhinovirus, metapneumovirus, one thing we know more and more. 10, 15 years ago, we just emphasized on influenza. But now we know that amongst the young and the old, we do see cases of respiratory attacks. The young gets hit quite badly, so do the elderly. But the problem is the elderly, a lot of them die. When they die, the graphics are missing. I'm really sorry. I'm anti-Apple now. My graphics are missing from this slide. Okay, so many people do die, especially the elderly, from these illnesses. So COVID-19 in Hong Kong, the cases are coming back. We are actually in the midst of a new COVID spike, and among the elderly, we are losing them. This is RSV, hits the young much more than the older folks, but some of the older folks do pass on as well. Influenza as well, beginning and the end of the year, we see them, and they do cause mortality. Highest amongst the elderly as well. It's supposed to be an icon, but I'm so sorry that the picture is not coming out. Vaccines. Vaccines is something which I'm passionate about. And if you go back into the vaccines in 2010, 2010 was much easier. It was just only influenza and pneumococcal. And that's how I started my speaking circuit. But thereafter, in 2023, we have a lot more. We've got pneumococcals, we've got shingles, we have RSV, we have dengue, etc. Going forward to 2030, we are going to predict a lot more vaccines availability. I'm quite excited. We're going to have the combination flu and COVID. There's development for human metapneumoviruses, and perhaps all these will change. At the end of it all, we're trying to improve the quality and life of our elderly patients. So going forward, we're going to see a lot more. We have the live attenuated virus as one of the earliest versions. Then we have the inactivated virus, the whole cell pertussis. And then we have the subunit vaccines that became available. That's exciting. Hepatitis B with the virus-like particles, and then we have viral factors that are coming on board. But what's really changed very, very much was actually the availability of mRNA vaccines. With mRNA vaccines, we can quickly design one protein, one antigen of interest, put it up, and we actually generate it very quickly. No longer are we dependent on growing it out from cell cultures or from eggs or whatever. We can make an mRNA, which is very simple, with the same lipid nanoparticles, and thereafter generate the vaccine. It makes incredible sense, especially in a pandemic or epidemic setting, or even a new virus or new entity. So just a quick reminder, you do the injections into the arm, it goes inside, the lipid nanoparticle allows the mRNA to enter into the cell, and thereafter the ribosomes that transcribes and comes out. And as we all know it, it doesn't enter the cell DNA, it doesn't change the human DNA. And there are many, many roles and potential roles for it. It's exciting. You have a missing protein, get the mRNA to do the job. You have the mRNA, you know the antigen responsible for protection, generate it, and then it presents itself to the antigen-presenting cells immediately at the site. So there's potential use for cell therapy, genome editing, as well as other potential therapeutic products. The mRNA, in other words, we are able to code the protein we want going forwards. mRNA vaccines, we have a way of saying things in Singapore. Is it the same, or it's not quite the same, or is it quite different? We have the Pfizer and Moderna vaccines. Frankly, I like the fact that there are two mRNA vaccine companies. It allows competition, and then it allows comparison, and we get to choose what's better. So you have the Moderna on the left that's blue, the Pfizer-BioNTech on the right. Both of them are mRNA-based, but the sequence in the spike protein is slightly different. As you all remember, we actually go into heterologous boosting at one point in time for COVID. The lipid nanoparticle formulation is different, and that actually makes the changes in terms of the strength of the vaccine. And there's also mRNA content that's different. You have a 50 microgram versus 30 microgram. Remember, previously, it was down to above up to 100 microgram. So the amount of mRNA which you put in is different between the two. Does it actually make a difference? This is data. There are many, many slides on this. This is just a summary slide. This is a meta-analysis looking at 24 studies comparing Pfizer, BioNTech, as well as Moderna. One, unity. If it goes to the left, it means it favors Moderna. If it goes to the right, it means it favors BioNTech. And you see that overall, there's a tendency. There is actually an improvement of outcomes for the Moderna vaccine. We have low risk of infection. we have lower risk of symptomatic infection, we have lower risk of severe infection, hospitalization, and incredibly, 20% reduction in hospital mortality. So actually better outcomes, even both of them are mRNA vaccines. After 5 years, did Pfizer improve on them? Not that I'm aware of. But Moderna actually has changed the mRNA vaccine. It's no longer, going forwards, the new mRNA vaccine will be different from the previous one that was launched with COVID. Besides the amount, they've changed the target. This is the original target. You see 3 different colors. There's gray, there's red, as well as there's yellow. Sorry, gray, blue, and red. So what they realized that the gray component is not important. What you really want is the receptor binding domain, and you want the N-terminal, and you just need to do a linker to link them up together. So you don't need the gray component, you get rid of the stem, and what you have is just focused target for the immune system to learn to overcome. That's what they did, old, new, and they've changed it just only receptor binding domain as well as the N-terminal, and you ended up with a much smaller dose of mRNA. Smaller dose of mRNA, and for people who work in the labs, the amount of mRNA lesser, perhaps it's a shorter strand, perhaps it will allow better thermal stability. So immune focusing design, lower dose formulation, overall, does it give you the same protection in the elderly, in the high-risk group? So on the left here, you have the 10 microgram, the new strain, as well as the old strain, which is 50 micrograms. And overall, we're talking about very similar, if not better effects compared to the old COVID vaccines. Less mRNA is needed, so this is an improvement from the last one. Flu vaccines. All of us have played with flu, all of us are familiar with flu, and all of us struggle with the challenges of flu vaccines. There's a production time, especially if you're down to the egg base. For the cell base, it's a little bit shorter. So there's, you choose a vaccine, and then thereafter the flu season, and we know that there's a new strain of H3N2 that didn't help. Strain variability, they keep doing antigenic shifts, if unfortunately, or we can actually have antigenic drifts. And then strain specificity, we are only as good as what we train the body to fight against. If there's a drift and the shift, it will mutate, especially during the egg propagation, and hence the popularity of the cell-based cultures. The egg propagation does change the structure of the epitope that's presented. Immunosenescence, this is where Sanofi was smart. They came out with a high-dose flu vaccine, and with that, we actually have better data and better outcomes for the elderly. So the DNA, what they've done was, be it the DNA or RNA of the known sequence which they want, in this case, it's actually the flu. You put in the mRNA, put in your lipid nanoparticles, as well as the buffer enzymes, and thereafter you'd have the valve in the vaccine, and you can do injections to the person. So if they've done this for influenza, for Moderna vaccine, and the advantages were precision antigen engineering. The egg doesn't go into post-processing, it doesn't change the antigen. You actually have endogenous protein synthesis and glycosylation, which means it is exactly what you want it to be, and it is right at the cell next to the antigen-presenting cells. The antigen will present directly to the immune system and becomes highly reproducible manufacturing process because they're just doing the mRNA manufacture. And at the end of it all, you have a nanoparticle system that's free of antibiotics, free of adjuvants, and it doesn't interact with the human genome. So this is pretty much similar to what we saw earlier. You have the mRNA going into the system, encodes, extracts out, you have your hemagglutinin glycoprotein that's expressed. It overcomes many of the limitations. Specific antigen, what you give is what you get. There's no requirement for egg-based or complex culture system. You can change it very quickly. The production time is much shorter. And very importantly, it has superiority for the elderly. Because Moderna didn't target it against the standard flu. They held themselves to a much higher standard, and they held it against Fluzone, which is a high-dose influenza vaccine by Sanofi Pasteur. This is the geometric mean titers for Fluzone, adults more than 65 years old. This is Unity, superior in the H1, H3N2, B Victoria, and V Yamagata. The seroconversion rates are also much better compared to the Fluzone. Superiority was demonstrated for all the 8 immunological endpoints. The GMT ratios as well as seroconversion rate differences were much better. Vaccine effectiveness, efficacy is favorable for the mRNA vaccine. Again, here, that's the mRNA, 1010, against the standard flu. Here you go. It is actually superior down here against for all strains. You might catch me and say that the flu B didn't quite cross the line. Challenges in flu vaccine and COVID vaccines. The existing vaccines has its problems. It's suboptimal. I love the flu vaccine, but I think we can improve on it. And mRNA actually helps address the current problems until we get the universal flu vaccine. But that's a talk for another day. Manufacturing mismatch is an issue, but the mRNA vaccine can actually help it. Seasonal timing, in other words, mRNA can wait until the last few months before manufacturing, before production, before delivery. If at the end of the day we cut down the flu attacks we're going to cut down the stress on the public health system. Co-administration, I would like a COVID and flu vaccine together. So one shot deals with both, and this is coming up. This is coming up. The less clinic attendances there are, the better uptake of the vaccine there will be. And this is something which we talked about it earlier today, adult behavior problems. There's a thing concerns about vaccine hesitancy. We all need to learn to convince our patients better. There's a crowded schedule, less people, less visits required, they'll be better. So the other vaccines that are trying to do flu and COVID together, we have Novavax. That's the antigen-based one. But we have Moderna here as well, doing the mRNA. Down here, actually, I just want to show you this something. So the BioNTech, they tried it, but haven't been quite successful. You do have Sanofi here, and GSK is hopping onto the mRNA vaccine. That's the powerhouse of vaccine companies previously. They're going into the mRNA manufacturing as well. mRNA 1083, this is actually 1083. So you actually have a flu and COVID combined together. It's a combination. And what they do is they do what the WHO recommends and then follow with the SARS-CoV strains as well, all into one dose, 0.32 mils, and it's recently approved by EU. So we have it available in approved in EU combination. So at one shot, one vaccine, one visit, you actually have protection for 2 influenza and SARS-CoV. So there you go. You have this is 1083, which is a combination versus the high-dose flu. And you have the results is actually at least as good as, if not better. Interestingly, the SARS-CoV-2 as combi administration was much better, superior. And this is for more than 65. 1 visit, 2 vaccines, and 1 convenient time. Geometrical titers are also much better as well. There's the combi, and there's the standard dose, mRNA. It works out quite fine. And this was 50 to 64 years old, is actually superior. Side effects. I think everyone wants to know about the side effects. I'm going to go through this slide quite slowly. No, it didn't quite work. Okay, this is separated, and this is combined. The reaction is very similar. Majority are grade 3 and 4. Down here, majority are grade 3 and 4. Very mild erythema, very mild risk of swelling. You may have some axillary tenderness or swelling. It's very similar to placebo, so that's expected. So the reported side effects were grade 1 and 2. Overall lasts about 3 to 4 days, no serious side effects. And this is the local, these are systemic adverse reactions: fever, headache, fatigue, myalgia, arthralgia, nausea, vomiting. It's pretty similar. In fact, there may be a little bit more, but all of them are grade 3 and 4, nothing of big concern. Reported side effects are it's supposed to be grade 1 and 2 with average duration of 3 to 4 days. Next, the RSV vaccines. All of us are familiar with the two RSV vaccines. One's from GSK, one's from Pfizer. The biggest trouble which I have as I administer the RSV vaccine is, when do I give the next dose? GSK has published the data. When you do a revaccination, you don't actually have the rise in antibodies. Pfizer has never published the data, but Pfizer has the data as a poster, and it shows that when you actually do a revaccination, it doesn't quite help. And the main reason is that the body immune response is a little bit different with the second one because it's neutralizing against something other than desired antigen. So what Moderna has done is they've done an mRNA RSV vaccine. Again, it's the mRNA sequence of the pre-fusion anchored RSV F glycoprotein, goes inside, mix it together with the stem, and what happens next is that my graphics are all coming out wrong. So you have the rise in antibodies, and thereafter it falls, and then when you do those again, it rises up nicely at very, very similar levels to what it was before. And this was 1 year. So there is revaccination data for mRNA. So the biggest question in everyone's mind is, if I can't revaccinate with GSK, can I do a heterologous vaccination? Can I do a GSK first, followed by Moderna? Can I do a Pfizer one, followed by Moderna? And this was published, this was released very recently in April. This was in ECAC. And when you have the pre-revaccination, post-revaccination, heterologous revaccination, primary, you end up with a similar result. So Moderna is presenting itself as a suitable revaccination vaccine, if not just a primary vaccine for RSV. And that way, we can actually potentially may have good protection for RSV going forwards. Vaccination, well, not vaccines. Vaccines don't save lives. We have, frankly, very great science, and things are improving, but what we lack is actually getting the vaccination across. So convincing our patients, overcoming all the vaccine hesitancy, and overcoming all the misinformation is truly important. Science is there, it's growing. We need to improve vaccination rates. So great vaccine, we are improving. In fact, besides mRNA, there are still other protein-based vaccines that are coming out into the market. This is great. More options, more choices. We need to have policy and recommendation based on evidence that helps. We need to identify the target population, cost effectiveness, operational as well, how to deliver it to them. And very importantly, public acceptance, potential acceptance of the vaccine, the physician and healthcare attitudes need to change and improve. If all these come into place, we have for ourselves a much greater, better looking world ahead of us. Now, this is something which I love to talk about. And this is just to straddle a little bit across to the elderly. The next talk is actually on elderly, but the speaker is not here, so I'm going to touch a little bit. All of us, we hope for our parents, we hope for ourselves, that we go down the healthy aging slope. Healthy aging slope, that means we don't fall sick, we don't fall, we don't break a bone, break a anything. And we just go into the sunset slowly. But unfortunately, many of us will have dips and dips and dips and dips. The dip occurs with a silly cold. The dip occurs with a silly influenza, COVID, RSV, or whatever. And when after the illness, they never quite recover. They are always a little bit further off. So for the elderly, the most important thing is actually preventing that first fall, that first illness. And vaccines offers a great benefit for that. And when we actually deliver a good vaccine and we deliver it to the patient, we're generating better karma for ourselves as well. So the other thing, just to end up with, Paul said that should I have been an ID physician? So if I became a family physician and I gave a lot more vaccines to everybody I would have generated a lot more better karma for myself. The ID physician who treats every complicated pneumonia there is. Okay, so very, very quickly, if this is something which I do with many of my doctors, and what I do is you imagine yourself someone you love. This could be an aunt, your father, your mother. Put it at the top of your head there, seated in front of you. Ask yourself, is that person truly protected? And is he going down the blue slope or is he going down the red line? If you feel that the person is going through the healthy aging, lift up your left arm. If you feel that that person you love so dearly is going down the dips, they put up your right arm. You want to do this exercise? Okay, never mind. So at the end of it all, we can always bring them back to health again. Besides eating healthy, stop eating wonderful food in Hong Kong, and exercising and getting your vaccines, we can bring them back to good health, preventing their first fall. Okay, right. I think I'm going to stop there. Yes?
Thank you very much, Ho Nam. Let's give him a round of applause. So we have quite a few questions online, but is there anyone in the room with a question first? Ariane has some mics. Okay, let's start with the online questions. So there are two questions which I think you kind of answered. First one is on comparing the RSV vaccines. So the questioner wants to know, in terms of effectiveness, whether there's any solid data comparing them.
No, not yet. We don't have solid data, but it's predicted to do just as well.
Right. Another question on comparisons. So this comparison is with the topical influenza vaccines versus mRNA 1010. That
So
one,
this
I
will be
think,
intranasal, isn't it?
that's right.
I don't think there's any comparison for that for the moment. But I think it's a great idea. It's a great suggestion. Having said that, the intranasal influenza vaccines haven't delivered yet against the standard dose influenza given intramuscularly. So I've always thought that the holy grail was the intranasal flu vaccine, but I've not seen great results delivering that holy grail.
Well, this whole question of mucosal immunity, I think, is
correct.
something that comes up
Yeah.
quite a lot. Okay, there's another question, and this is something along the lines of the universal vaccines. Do you think it will be possible to develop a vaccine to a conserved region in COVID or influenza or RSV?
The answer is yes.
Okay.
It's an
That's very
absolute yes.
optimistic.
In fact, I mean, just it was just a few months, or was it a month ago, with the universal COVID vaccine that's designed by AI's help. I'm really looking forward to that. We may need a few repeated doses every now and then, but one is almost like Lord of the Rings story. One vaccine to rule them all.
Don't we all hope for that? Yeah. There was another question earlier on related to the slide which you showed about the comparison between BioNTech and Moderna for COVID. And the questioner says he understood that there was a trade-off between effectiveness and safety. So were there more AEs with the mRNA because of the higher dose? You know, this everyone talks about the Moderna arm.
The Moderna arm, many people gets a little bit more reaction. But the older you are, the less reaction you're going to be. And once you do have a reaction, it tends to occur in the first 1 to 2 doses. Third dose, the risk really comes down quite tremendously. There Okay, for the at-risk population, the elderly, the answer is no. I don't think it really makes a significant difference. And the way I do it for my patients is, oh, you got a reaction, great, congratulations, because you have great immunogenic response to it. So there is a potential trade-off in that sense, but the numbers are very, very few. Number two, but the trade-off in terms of effectiveness, 20% reduction in mortality. That's incredible. A little bit of ache, 1 day of ache, 1 day or 2 days of ache with a 20% reduction in mortality. I'll put that ache up first.
Okay, next question is, there are two questions actually from Agus Siahura Rahman. First question is the old and new mRNA vaccines in pregnancy. What do you think in terms of benefits and risks, especially in the first trimester?
Okay, so I think it should work just as fine. I haven't seen the data for pregnancy, but I expect it to work the same. There shouldn't be any problems. That's number one. Number two, I would love to give the pregnant women their COVID vaccines, but many women will scream at me and say no. Let's be very frank and honest. Number three, if I have a choice to give it, actually I'd rather give it in the second 20 weeks rather than the first 20 weeks. So it literally is a buy one, get one free. One for the mom and free for the kiddo.
Yeah, I think that's what most of the obstetricians would rather do.
Yes.
And I think there's a couple of reasons. One, obviously, in the first trimester, they don't like giving anything to pregnant women. And number two, there is a significant rate of fetal loss in normal pregnancies. And you don't want to get blamed for it, for giving a vaccine which is not mandated. So you tend to give second trimester.
I tend to give second trimester.
Yeah.
In a way, it is inside guidelines. So when obstetricians don't give it during guidelines, it's
Right.
against guidelines. But very few, unfortunately, the pickup is very, very sad and pathetic.
Okay, last question. So do you think that there's any role for this prolonged, broadly cross-protective mucosal immunity by, say, so this is the opposite of one vaccine to save them all.
Yeah.
If you give sequential vaccines, whether you're going to achieve...
I think that would be really very nice and it's very sexy. But until that day comes, I will continue to pine for it.
Okay, on that note, thank you very much, Ho Nam.
Thank you.
So I'm going to introduce our next speaker. So the next speaker is Professor Colin Russell, who will be joining us virtually from Europe. He is the Professor of Applied Evolutionary Biology at Amsterdam University Medical Center, and he'll present on leveraging combination vaccines for global health impact. Professor Russell, can we... Ah, there you are. Well done.
Let's give.
Excellent. Let's do it.
Perfect. So I assume that you all can see me. And if we
Yes,
can have
we
a
can.
little audio check to make sure that you can also hear me.
Yep, we can hear you.
Very good. Then I'm going to share my slides and start my talk. So thank you all very much for giving me the opportunity to speak today. And I'm sorry that I can't be with you there in person. I was asked to talk about combination vaccines and their potential impact for global health. And it's always difficult to follow a data-driven talk like the one that we were looking at previously. And here I'm giving more of a perspective on what I see the opportunities and the challenges are for these vaccines. And I think it would be easy to look at this talk in hindsight and say, he's very skeptical about combination vaccines. And that's not the viewpoint that I want to give. I think right now we're in an exciting period of development where there are opportunities, but there are also very substantial challenges. And I'm trying to give an objective and nuanced look at where things currently stand. So these are my disclosures. I have received support from the sponsors of this meeting and from other pharmaceutical manufacturers, but I think if we look carefully at that list, we see that there are both combination vaccine manufacturers and non-combination vaccine manufacturers on the list. And so
Sorry, we've lost your
that cancels
slides. Can you share your screen again?
Sure, 1 second. There we go.
Oh, you came on and then disappeared. Ah, there you are. Okay.
Okay, Perfect. You see my slides now?
Yep.
Okay, perfect. Thank you. So what I'm going to cover today, I'm going to talk first about the case for combination vaccines and why they have worked well. Talk about the adult respiratory virus landscape and why it is potentially different and more challenging than the cases where combination vaccines have already worked. Talk about some of the logistical and supply chain obstacles. Talk about the evidence in terms of where things currently stand, particularly in the context of a newly licensed COVID influenza vaccine in Europe. Talk about some of the policy, NITAG, and health economics issues, and then conclude with talking about what we still need and the remaining evidence gaps. So first, the case for combination vaccines and why they've worked. So I think many of you have probably come across this quote previously from Bill Hausdorff, who's the director of PATH. And it's basically that the history of immunization is a history of combination vaccines. And in principle, it is a beautiful idea. And in fact, combination vaccines are already widely used. I mean, they are the basis for childhood immunization programs. And here we're looking at a little bit of a timeline of some of the major developments in that space. And I don't think for this audience, we need much of an introduction on how successful these programs have been. But in terms of why they have been successful, the antigens that are in these vaccines are stable. There is no need for reconsideration of their formulation or for them to be reformulated with any degree of regularity. The target populations for these vaccines generally have, or ideally don't have, any pre-existing immunity to interfere with. And I think the other element of this that's really important is that there are predictable and widely agreed upon non-seasonal dosing schedules. So in general, despite vaccine hesitancy and vaccine skepticism in some parts of the world, in general, we agree that childhood vaccines are important and we agree on what children should receive. When we think about transitioning this, though, into adult respiratory virus vaccines, these features don't necessarily apply. And so why are adult vaccines different? And I think for thinking about that, we need to focus on exactly what we mean when we're talking about combination vaccines and what we might expect to see included in them. So we're talking about really the big three of adult respiratory viruses. And I say that I mean specifically influenza, RSV, and SARS-CoV-2. The burden of these is familiar to all of you. And I think some of the key differences, though, are in the antigens themselves. So when we look at flu, I think all of us are very familiar with the fact that flu evolved rapidly and that we need to perform vaccine strain selection at least twice per year. And in the vaccine space, for flu specifically, we already have licensed high-dose and adjuvanted products, as well as a whole plethora of standard dose egg-grown vaccines. Now, modest vaccine effectiveness in older adults tends to be relatively modest, but it depends on what our baseline expectation is. If we're hoping for measles-like vaccine effectiveness, that is probably not realistic for adult respiratory virus vaccines. However, when we think about sort of a 40 or 50% vaccine effectiveness, that is actually a substantial amount of protection. When we look at RSV, RSV has a relatively stable antigen, and it looks like a single formulation is possible. And at least early data on these vaccines suggest that there's good efficacy across the first season, and it might be possible to have multi-season protection without any boosting. SARS-CoV-2 is a guess as much as anybody is concerned. The spike protein is highly mutable. Vaccines need to be updated regularly. And there are a variety of licensed products, both mRNA and protein-based. But these formulations are all substantially different from one another. Now, when we look at the asymmetry between pediatric vaccinations and adult vaccinations, we talked before about the fact that childhood vaccines are basically antigenically stable. The hosts are immunologically naive. There's only a single formulation needed, a synchronized dosing schedule. And there's also decades of real-world combination data that help support their use from a policy perspective. When we look at the combination vaccines for adult respiratory viruses, well, for influenza and COVID, we likely need to change those antigens at least every year. Recipients carry decades of prior immune history, and we know that affects vaccine effectiveness outcomes. Right now, at least in the context of COVID and influenza, it's not clear that these epidemics are sufficiently synchronized temporally that it is necessarily the best idea to give these vaccines at exactly the same time. Though we'll come back to that more later in the context of co-administration. One of the risks that I don't want to dwell on too much because I think it's readily fixable is the potential for antigenic interference between different components of these vaccines. On the one hand, this is a reasonable concern, and it's something that needs to be meaningfully interrogated because rational skeptics could raise this as an issue. But at the same time, we already do, to a certain extent, see these issues with seasonal influenza vaccines that we already have, where we already give 3 influenza vaccine strains in one vaccine, which in and of itself makes it a combination vaccine. And interference doesn't seem to be a major issue. So the extent to which that issue exists across other viruses as well is probably something that can be ameliorated. Now, when we look at RSV, the question becomes a little bit different. And it's one of the reasons why I think that many manufacturers have been reluctant to include RSV in combination vaccines so far, which is that it's not clear that RSV will need an annual booster. That complicates the concept of giving a trivalent combination vaccine that includes influenza, SARS-CoV-2, and RSV. When we look at the potential logistical challenges associated with routinely delivering combination vaccines, there are potentially misaligned update cycles. So speaking in broad brushstrokes here, because I know this isn't particularly exciting, but it is something important for us all to bear in mind, is that influenza and SARS-CoV-2 do need to be updated regularly. However, they might not need to be updated at the same time. So if we're selecting for vaccine formulations that are challenging to update or that require multiple different updates across different vaccine components, that adds substantially to the complexity of vaccine production and the logistics and timelines. And I mentioned before that the RSV component, based on current data, doesn't necessarily suggest that we would need to be vaccinated against RSV every year. It complicates the overall logistics of vaccine design because then we have a question about whether RSV should be included in routine combination vaccines or not. But that's something we'll come back to on subsequent slides. There's also potential supply chain risks in the context of producing combination vaccines. And there, the real headline concern is that a manufacturing or regulatory failure in any one component halts the entire production line. So this is an issue that will be very familiar to influenza vaccine manufacturers, where you've got to get at least 3 strains in the trivalent formulation working and efficiently delivered. And in the context of a quadrivalent vaccine, there were 4 strains. And it's usually easy to get a couple of those, and sometimes all of them to work well. But if one of them presents a challenge, it can delay the delivery of the vaccine product. In the context of combination vaccines, this only becomes more complicated when we think about adding different antigens for different pathogens. This creates risk, and risk is a manageable component, but it's something that we can't just go in assuming that everything is equal and fine. So in the context of a combination vaccine, a single quality failure halts both products. In terms of supply chain disruption, one antigen component affects the entire product, and this results in an associated manufacturing complexity. When we look at co-administration, a COVID-19 batch failure doesn't affect influenza vaccine supply. And that sort of flexibility is something that's important. We will come back to more in a moment. Independent manufacturing supply chains for separate products effectively builds in redundancy. And a shortage of one component, not necessarily a delay or a production issue, but a shortage of one component doesn't delay vaccination against the other components. And so there are potential still advantages to co-administration that go beyond the convenience that might be associated with combination vaccines. Looking at where things stand today, because on the one hand, there's a lot of buzz about combination vaccines. At the same time, there's only one currently licensed combination product, and that's a relatively recent development. So in terms of where things stand with co-administration, and there I specifically mean separate injections on the same visit. There's good data for RSV and influenza, where there's only modest titer reductions associated with co-administration. Of course, this needs to be watched, but it's not concerning. In terms of RSV and mRNA influenza, there's non-inferior immunogenicity, and second restraint responses have been variable but similar. And in terms of triple vaccination, here, the only product that's actively been tested, there were no adverse signals, but of course, trial design was hypothesis testing and not confirmatory, but initial results look positive. So the headline there in terms of combining things versus co-administration definitely works. Now, for combination, so these are fixed combination formulations. The exciting one, of course, is Moderna's product Encabriax. So Encabriax was licensed in the EU in April of this year, and it's a flu plus COVID mRNA vaccine. And it is exciting because it shows non-inferior and statistically superior influenza responses compared to other licensed products. Now, one of the important things is that this vaccine was approved on immunogenicity. There is no vaccine effectiveness data available for that yet. And that's going to be an important space to watch as we move forwards, because one of the primary issues associated with that product is that it is expensive, and it's going to need to outperform existing products on something that goes beyond the convenience of just giving one vaccine. When we look at other combination products that have been tested, Pfizer BioNTech have a flu and COVID combo, which met its non-inferiority for COVID, but and for influenza A, but it failed on non-inferiority for influenza B. Novavax and Sanofi have a COVID influenza vaccine in trial. It's currently placed on hold, or was placed on hold in 2024. And initial data is positive following a 2025 reopening of that trial. And then Sanofi also has a flu plus COVID combination that's based on a different formulation. And that is currently still in testing in phase 1 and phase 2. So there is a lot of development going on in this space. But in terms of evidence of superior effectiveness or improved public health outcomes, that still remains to be seen. So one of the things that we need to talk about very critically is what Encabriax did show in terms of their effectiveness and what they didn't show. So it was non-inferior and superior for H1H3 B Victoria versus comparators. So as a flu product, it generated superior immunogenic responses compared to a standard dose influenza vaccine. And it also induced superior COVID-19 neutralizing antibodies relative to a standalone COVID vaccine. Those two findings alone are striking, and they give reason for a lot of enthusiasm for what this product might be able to do in the long term. It has now been licensed in Europe, and this fall, we will see the first sort of pathways to uptake and potentially get early readouts on vaccine effectiveness. But that vaccine effectiveness is one of those things that has principally not been shown so far, since it's only been approved on immunogenicity cohorts. The flu component is not separately, has not been separately EU approved, and the FDA has deliberately withdrawn consideration of the combination product until there is empirical standalone data for the flu product. One of the things that's important is that there's no immunocompromised data on for the use of this combination vaccine. It was not included in the phase 3 trial, and it's an explicit EMA label gap. So that's something important, particularly when we think about the people who potentially most need to receive these vaccines. There is an annual update mechanism that has been proposed, and if viable, it should be fine, but it's untested in practice, and so there's something that remains to be seen. Now, one of the areas that I think we have to talk about with extreme care and nuance is the issue around reactogenicity. For many of us who have received mRNA vaccines, the reactogenicity profile is unpleasant. We typically feel sick for a day or two. And this is unfortunate, but it is one of the realities of the current mRNA vaccine formulations. Now, in the phase 3 trial that's based on Encabriax, there was reported difference between the combination versus co-administration routes of 83.5% versus 78% in terms of adverse events in people over the age of 65 for the combination versus the co-administration routes. It's roughly 5% difference. And with adverse events being that common, 5% difference is not meaningful. And in the paper, that's used as the basis to say that these vaccines have very similar reactogenicity profiles. But a key thing to consider there is that both arms of the vaccine trial contain Spikevax, an mRNA COVID vaccine, which drives approximately 80 to 90% of adverse events when administered alone. So what this means is that adding mRNA flu to mRNA COVID barely increases reactogenicity. The change of the delta of 5% is really a floor rather than a result. The reality is, particularly given what we see in terms of COVID vaccine uptake globally at this point, where it has declined substantially, is that a relevant comparator is giving an inactivated influenza vaccine alone compared to giving a combination mRNA vaccine. There, if we look at Fluzone HD, there is approximately a 21% adverse event rate, which is very, very different than an 83% adverse event rate seen in the context of the combination vaccine. So the true gap in the reactogenicity profile between co-administration and a combination mRNA product is potentially very much larger than the 5% that's been reported so far. Now, this isn't necessarily damning, and I think that's worth being clear about. But at the same time, in places where vaccine hesitancy is common, where there is tremendous vaccine fatigue, high rates of reactogenicity will decrease uptake. And that is a challenge for all mRNA vaccines, regardless of whether they are single pathogen or combination. And it's important that we take those into account when we're making public policy decisions moving forward. Now, when we look at policy, NITAG, and the health economics of these vaccines, co-administration is still the pragmatic near-term win. So when we look at Combreax, which is flu and COVID, versus co-administration, here thinking critically about flu, COVID, and RSV, since all three are important. Combreax does deliver flu and COVID in one vaccine. Co-administration of all of these separately would be 3 vaccines. But even if we consider RSV to be an important component of the vaccine schedule, Combreax of flu plus COVID still requires 2 administrations once we include RSV. Combreax doesn't cover RSV, obviously. Resolving annual complexity. In the context of flu and COVID, they are a very convenient marriage because both of them need to be updated frequently. Not necessarily at the same time, though. In the co-administration route, the independence of vaccines allows for independent update schedules, which simplifies the process. Immunocompromised data is currently absent for all licensed combination products. However, in the context of flu, COVID, and RSV independently, they're licensed for immunocompromised use. Regulatory approval, currently, Combreax is only going to be available in the EU. FDA is pending, and there are other considerations happening in other parts of the world, but currently flu, COVID, and RSV are available certainly in all regions of the world, if not most countries. When we look at near-term availability globally, that's EU for combination vaccines, but co-administration is now possible everywhere. So when we think about global policy, there is naturally excitement for a combination vaccine, but co-administration remains the more viable route currently. But here are a couple of things that have to be considered in the context of NITAG and cost effectiveness. And the first one I'll skip over since I've already talked about it a fair bit already, but that is if there's a person out there who needs a flu and COVID vaccine and RSV, a combination that includes all 3 might not make a lot of sense in the context of what RSV recommendations might end up looking like, where RSV vaccines don't need to be given every year. And this creates the broader challenge of the fact that flu, RSV, and COVID recommendations are not coterminous in certainly most countries in Europe, but I think it extends to most countries globally, where there are different eligible age and risk groups, and the fixed combination may require changes in schedules or very narrow recommendations. In this way, parallel monovalent inventory almost remains mandatory regardless of whether or not combination vaccines are widely introduced or not. The second issue is one that's very practical and grounded in health economics, which is the likely premium price of combination vaccines and the yet unproven incremental benefit. So these vaccines have been licensed on immunogenicity, and they do show superior immunogenic titers. But whether this translates into fewer hospitalizations is genuinely unknown. The incremental benefit could be real, but it's currently unproven. The price for these products is almost certainly going to be high, and bundling does not necessarily lower the cost of goods. But beyond price, given that we don't know much about its real world and clinical efficacy, the value is yet to be established. But even once benefit is established, there will be a fixed willingness to pay premium for a premium price that simply, well, let me start that. My apologies. There's going to be a fixed willingness to pay a premium price that simply raises the cost effectiveness age threshold. And so this is something that's very clear in the UK right now, where they have narrowed the eligibility for people for COVID vaccines based on the fact that those vaccines are expensive. And so JCVI in the UK has narrowed the COVID recommendation to people who are 75 plus, precisely because that's where their cost effectiveness threshold lies. And when we think about a very, very expensive combination product, and that is speculation on my part, to be clear, the cost effectiveness threshold and the age groups that will be, those vaccines will be made available to are likely to be very small. And we also need to think critically about who will actively choose to receive these vaccines. When we look at vaccine uptake in older adults, so people age 65 plus, and we look at influenza versus COVID by region, here just comparing the United States and Europe. So in the United States, influenza vaccine uptake is about 44% versus 31% for COVID-19. When we look at the EU, it's 32% for flu, but there's a tremendous amount of heterogeneity across different EU countries. But there's near homogeneity in terms of the very, very low uptake of COVID vaccines. This creates a real challenge because we then have people who are either not being offered or who are actively denying COVID vaccines. Already mentioned briefly before that in terms of JCVI's recommendation, this is for the UK, COVID is restricted to over 75s, care home residents, and the immunosuppressed only. 65 to 74s were removed just this past year in terms of their vaccine eligibility for COVID. Their standard cost effectiveness methodology applied at £25 per dose. If the combination vaccine price is higher than that, then we're going to see a further narrowing of any sort of more expensive vaccines. And so that means that the target population in terms of who might actually receive these vaccines is potentially very small. When we look at the value proposition, the compliance benefit requires that both vaccines are recommended to the same person simultaneously. And as the UK example shows, that's increasingly rare, where people 65 plus are all recommended to receive influenza vaccines, but not necessarily for COVID vaccines. But then there's also patient acceptance themselves. And I think this is something that is reflected in terms of vaccine uptake that likely exists in the audience, which is there are plenty of people who are flu yes, but COVID no. And here, a combination product, if it's the only iron on offer, offers a binary choice, which is to accept an unwanted COVID component or lose flu too. And on the one hand, there are many arguments to put forth that say that combination vaccines might increase vaccine uptake. But I would also suggest that we consider that combination products also risk that we decrease vaccine uptake, either because of side effects or because of components that patients themselves don't deem risky. So when we talk about the evidence that is still needed, we need vaccine effectiveness data for combination products. It has not yet been measured, but when it comes, we can hope that it will be encouraging. I think, like all of you, would love to see more effective vaccines that reduce the burden of respiratory viruses, but we need to see them in order to believe it. Annual update mechanism. There's at least a plan in place for the EMA and the EU. This will need to be tested this year, but also we need to see how this looks when we update it on a global scale. We need to see how these vaccines work in immunocompromised and frail older adults. These are currently not included in trials that have been published to date, and the EMA label explicitly notes that there's no data for immunocompromised people. This is an important gap in our capacity. And then finally, RSV is not currently included. The triple burden remains unaddressed. And if we think about really the breadth of possible major respiratory virus challenges, the opportunities for a combination vaccine, and for reducing the number of vaccines that people receive, a triple combination makes a lot of sense. And yet, for all of the reasons that we were outlining before, that combination is not a trivial thing to pursue. So, conclusions. And so really, it is exciting that there is now a licensed flu COVID combination product. And the freeze-3 data is very encouraging for what its potential might look like once it's actually widely used in humans. However, we do need to see what it looks like when it's widely used in humans and to look at the incremental benefits of these. And for the time being, co-administration remains the more viable and flexible route. But then I want us to think just for a moment that actually, on the one hand, combinations and compliance, they solve potential issues. However, immunosenescence, imprinting, and prior vaccination history all have incredibly important impacts on vaccine effectiveness, and they remain the most difficult and unsolved problems, both for combination vaccines and monovalent vaccines alike. So these combinations of challenges mean that we need to potentially temper our enthusiasm for combination vaccines. There is a lot of potential, but we face a lot of challenges. But thank you all very much for your time and attention.
Thank you very much. So any questions from the audience? So we have a couple of questions from Prof. Agus. And the first one is about in vitro testing. So is there any in vitro data that given combination antigens presumably is going to interfere with replication in vitro in cell lines? So in other words, if you give, yeah, sorry, go ahead.
No, please go ahead.
Yeah, so I mean, I'm interpreting the question to mean that, most of the studies have been done presumably in humans, but is there in vitro data to suggest otherwise?
No, I mean, for the moment, the best available data is from humans, and I'm not aware of anything beyond that.
Okay, good. Then the second question which he has is related to this issue of reactogenicity. So if in a combination, the more reactive component of the combination vaccine, will that affect the immunogenicity of the other component? So in other words, if in a reactogenic vaccine, you're going to get a lot of pro-inflammatory cytokines, is that going to affect the immune response? Or do you know from the studies whether there's any correlation between the highly reactogenic vaccines and lower immunological responses?
And this is a challenging issue, to say the least, because it's driven by a complicated phenomenon where the reactogenic component of these vaccines is the lipid that the mRNA exists in when it's being delivered, and rather than the antigen itself. So the antigen itself, it works very well, but we have to deal with the reactogenicity profile of the lipid delivery mechanism. There is some evidence to suggest that high levels of inflammation potentially could either help or hinder an immune response. But I don't think that there's good evidence to suggest that one way or the other in terms of whether or not that inflammation is driving better outcomes or worse outcomes.
Thank you very much. I think you've given us, John has a question. Yes, please, John.
Thank you very much. Wonderful presentation, really interesting. I was thinking, what would you think mechanism of what we call mix and shoot approach rather than a combination all-in-one approval? Let's say you approve 3 separate vaccines, right? And then at the time of immunization, you mix them and then inject. So then you have the 3 vaccines all approved.
So that's like it's like co-administration, though. The co-administration in the same tube.
Co-administration in the same tube, same noodle, same site.
Exactly. Yeah. Exactly. So I mean, funnily enough, you mentioned that. I took my cat to the vet yesterday. It's 6 months old and had to get booster. And the vet did exactly that. She only wanted to give him 1 needle, and so she combined 3 vaccines in 1 needle. Now, I don't know of any good data or guidelines for clinical practice that would allow us to do that in humans in a way that has been tested or validated. And that's the thing that I think we have to be careful about, which is the idea itself is sound. And it is an excellent opportunity to only give 1 injection rather than 3. But I think we would want to be careful in evaluating what that actually looks like in practice, if not only because the quantity of vaccine delivered to that point would be fairly substantial, and the local inflammation and injection site pain could be substantial.
Thank you very much. Oh, there's another question. Given that intramucosal flu vaccines have been developed, how does the route of administration complicate administration of combinations? So I guess the question is, you've been talking primarily about intramuscular, but do you think there's a future for intranasal combinations?
Honestly, I think it is difficult for me to overstate the promise that I think exists in the context of intranasal vaccines. And it's twofold. So one is the sort of obvious practical element, which is that people don't like needles. And if you could just get a little spritz up your nose instead, that would be nice. But I think the more exciting element of all of that is the fact that if we want to think not only about reducing severity of disease, but potentially preventing transmission of disease, the only way to do that is to ensure that there are good localized immune responses in the primary sites of infection for these viruses. And so that's where the intranasal component comes into play, because there's lots of evidence to suggest that the intranasal route of administration gives better IgA responses on mucosal surfaces. And these are the responses that are the most likely to provide protection against infection. These vaccines already exist for flu, and they're widely used in multiple countries.
Yep.
One of the challenges that they face is the fact that older adults tend to respond strongly to the vaccine to the point where it neutralizes the vaccine before they generate new immune responses.
That's right.
This is a challenge that needs to be overcome.
That is a huge challenge, and I think that's a good note to end the session. So thank you very much for your very thought-provoking presentation, and thank everyone for listening in. So we're going to take a break and reconvene at 10 minutes to 4. Thank you very much.
Thank you.